Today's seminar is going to be a little bit different than your usual CME seminar in that it's not really going to touch on the day-to-day kind of routine of an ENT doctor. But I'm hoping to appeal to your inner biologists and to convince you that you can turn to zebrafish as an animal model, as an ENT doctor, when you come up against research questions that you'd like to address in a quick and sort of comprehensive manner. So today's objectives include learning the basics about zebrafish, and then I'm going to give you a possible case scenario as an ENT doctor where you might turn to zebrafish as an animal model to study human deafness and even vestibular disorders. Okay, so work with zebrafish began in earnest in the 60s when George Streisinger, who was famous for his work on bacteriophages, decided to do something really different when he moved from Cornell to the University of Oregon. And he was always a hobby aquarist, and so one day he went to the local pet store and he bought all these different fish that he could find at that pet store, and he brought them into the lab and he worked with them for at least several months, if not longer. And through that kind of work, he discovered that the fish that was really the easiest to work with was the zebrafish. And they're called zebrafish because of the stripes along their body. So the zebrafish is really hardy and it's easy to breed. It's actually a tropical fish that originates in India or Bangladesh. Its natural habitats are in the slower moving parts of rivers or in rice paddies. Here these biologists are trying to catch some wild zebrafish and bring them back to the lab. So George he created the world's first fish facility in one of these old World War II military huts. So they had one of these extra ones laying around at the University of Oregon, and they said, here, do your zebrafish in one of these. So here he is inside with his many fish tanks. And it took him some years to publish something from this work. It took about 9 years before he had something. But it was a great paper that came out in Nature in 1981. So many of you probably think that Dolly the sheep was the first vertebrate to be cloned, but this is not true. It's actually the zebrafish, the very first vertebrate to be cloned by George. Okay, so the general outline of my talk will be reasons for choosing zebrafish. I'll talk a little bit about animal husbandry, point out some important genetic tools, mention some disease models, and then we'll move on to this possible case scenario. So why choose zebrafish? Well, one reason is that they are prolific egg layers. You can get up to over 300 eggs from a single female. And they are cheaper than mammalian models. For example, you can keep many fish in a small amount of water, and this fits very compactly into a small space. So a facility like this with this many racks can house well over 100 different mutant lines. And the food is relatively inexpensive, so the cost of this kind of operation is the cost of a salary of a technician and the little bit of money that you spend on the food. And that's really nothing compared to the amount of money that you would spend on an equivalent number of lines of mice.
Except it has to be in the basement with the floor drain, because if it's on a higher level, it floods the people below you? No?
Not true.
Oh, really?
So at OHSU, my fish facility was on the 8th floor of the building. And we never had floods. This is always some worry that, oh, all the water's going to spill out and there's going to be a huge, like 6 inches of... This doesn't happen. I mean, at best, you might get a crack in one of these and there's a drip. But think about it. How is it possible that these, clear acrylic tanks could cause such a huge flood. It's a very rare occurrence.
Flex seal works?
What's that?
Flex seal works?
Yes, I mean, you have to have plumbing, safe kinds of ways of sealing pipes and so on that won't lead to the death of your fish. So you have to be careful with, calling in any old plumber and make sure that he is using things that aren't toxic. Okay, but perhaps more importantly is that most of the anatomy and genes are conserved in zebrafish compared to other vertebrates. And this is despite having a common ancestor well over 400 million years ago. So fish are down here, and then we have all the other vertebrates that you're probably a little more familiar with in terms of use as animal models. But despite that huge difference, the basic anatomy is well conserved in that fish do have all the basic organs that we have. There's a kidney, there's a spleen, there's a pancreas, and so on. And in terms of the fish ear, there's no outer ear, there's no canal from the body, from the wall of the fish coming inward into inside, making a connection to the ear. This here is actually just a flap over the gills. So the ear would be sort of deep inside, encased in bone in this fish. The inner ear that they possess actually closely resembles the vestibular portion of our inner ears, in that there are 3 semicircular canals, all occupying different orthogonal planes, and there are various macular organs such as the utricle and saccule. And in zebrafish, they're using the saccule and also this other third macular organ, the legina, for hearing. So anything in blue is what each animal uses for hearing. So we can see that in the frog, they're also still using the saccule. But then in other vertebrates, it's an outpocketing near the saccule that has given rise to the cochlear structure. So they don't have a cochlea, but they are instead using macular organs for hearing. Now, most people don't work with adult fish. I think it's quite a challenge to work with adult phenotypes because it takes some time to raise fish. And most people are working in zebrafish labs are working with embryos or larvae. So these are 5-day-old larvae, and here is the inflated swim bladders. So they're anesthetized and they're just resting at the bottom of the slide. And then here's their eyes, and in between the swim bladder and the eye is the ear. And I think you can pick out these little stones called otoliths. And if you look at all the other ones, you can see where their ears and their stones are. So if we take a closer look at their ears, which are perfectly transparent, you can look directly into them. Even up to 3 weeks of age, you can still look directly into the fish ear and see the sensory neuroepithelium. For example, here are the 3 end organs in the developing semicircular canals. You can see the little kinocilia here. If we go a little bit deeper, we then see the macular organs with these large otoliths. So we have otoconia, but in fish and in some frogs, they have rather a large stone structure referred to as an otolith. So this is called the anterior macula, which is destined to become the utricle in the adult, and this is the posterior macula, destined to become the saccule. And in zebrafish, as well as many other fish species, but not all fish, have a series of bones that interconnect the swim bladder to a compartment associated with the saccule of the ear. And because of these bones, they're actually more sensitive to sound. So when sound travels through the fish body, it sets the swim bladder into motion, and that motion is translated by these bones to the ear. So they're a bit more sensitive in terms of detecting sound. So what is their sensitivity? What kind of frequency range do fish have? Well, earlier reports said that, well, they could hear up to 4 kilohertz, but actually in this 2015 paper, they did something akin to ABR recordings and found that they could see responses up to 8 kilohertz. And if they used loud enough intensities, they could even see responses to 12 kilohertz. So here comparing 108 and 12 kilohertz at different intensities.
Could that be from the lateral line as opposed to from the actual inner ear organ?
You know, in this case, there are electrodes in the brain, and they could be picking up something from the lateral line, but the lateral line in general is sensitive to much lower frequencies, like anywhere from 1 to 150 hertz is their range. Now, at first glance, you might think, oh my God, that's a really loud sound. Of course, you're going to get some response to that. But in these experiments, what they did was they heavily sedated the fish, and they no longer saw these responses. But you also should keep in mind that this is actually a different scale here. So this is underwater decibel intensity. And that's always relative to 1 micropascal instead of 20. And so if you take that underwater value to get to an estimate of what the comparable value would be in air, what you need to do is subtract that reference pressure of 26 decibels and then take into account that these speakers are underwater, so you have water, the density of water, which is more dense than air, and the speed of sound is faster. So you also subtract another 35 decibels to arrive at, you know, a value that's closer to 80 decibels, which is still pretty loud, but you can see that with 80 decibels, you're getting fairly reliable responses to a wide range in frequencies. If you look at thresholds, some of their best frequencies are here at much lower frequencies, between 600 to a kilohertz. You'll see thresholds around 150 or 105 dB, which corresponds roughly to about 45 dB in air, so a very loud whisper. So their sensitivity isn't that bad, despite having an ear that's encased in bone and having no canal to the outside of the body.
Why does the fish need to hear? I mean, you would think normally it's either to communicate with mates or to avoid predators or to recognize prey. Do they make utterances to each other? I mean,
for all 3 reasons you just named, that's why they need to hear. And there are definitely, zebrafish aren't vocalizers, but there are fish that vocalize to each other during the mating season or during certain times of the day. They'll croak. What they do is they use their swim bladders as drums. And they use the muscles to beat against the swim bladder. And they make all kinds of interesting noises that way. It almost doesn't sound like a real animal. I wish I had the recordings. I could have shared that with you. Sorry about that. They sound really bizarre. They're really low frequency, bizarre things that you might mistake for something else, but it's actual animals. The first time that people heard fish talking was during World War II, when they put microphones out in the Atlantic Ocean trying to listen for submarines, and they heard like the toadfish just doing this kind of noise the whole time. And they're like, what is this? They figured out that it was fish talking to each other.
Fish call one. Good morning.
Okay, so at this young larval stage, if this otolith is missing, the fish is incapable of hearing. So at young stages, this part of the ear is already dedicated to hearing. If you lose this otolith, then you lose vestibular function. So here's an example of where we took advantage of a particular mutant. It's a mutation in the autogelin gene that results in the lack of an anterior otolith. These fish can still hear because they still have that posterior otolith, but they have severe balance defects. And here in this test, we're testing for basically the vestibular ocular reflex, and it's completely gone in those mutants. Here's an example where this group physically removed the anterior otolith, sort of an acute kind of experiment. So in a normal fish, there is a slight tilt to the eyes, the normal resting position of the eyes. But if you remove both otoliths, then that eye position resets itself and you get a different angle to those eyes. And if you do unilateral removal, then you see the adoption of also other positions of the eyes. So this is kind of similar to the ice cube kind of issues that you might see in patients with vestibular problems. Okay, so another reason to use them is the ease of the genetic methods, and a lot of the basic genomic tools are in place. And people love them because of their very fast development and the transparency. The transparency was the biggest attraction to many people studying just basic development. So here's a movie of the first 18 hours of development where you can watch all the cell divisions and watch the morphogenesis. So here we're just having dividing cells, they're clumping up, but then they're going to start moving over the yolk cell, and eventually we'll see the head emerge here with part of the nascent eye present, and then some of the muscle somites here. So within 2 days of development, post-fertilization, all the major organs are in place, and by 5 days, the fish have inflated their swim bladders, they're free swimming, and they're even capable of catching prey and eating and feeding. And this is actually the stage that we use for our research. Okay, now just a couple of slides about basic animal husbandry. So many people ask me, how long do they live? They can live up to 5 years. At that time, when you need to cull your populations, the easiest way to do that is just to throw them on ice water. It's very rapid death. You can give them an overdose of tricaine as well. They mature within 2 to 3 months, so males are usually smaller than females and more colorful. Females usually have this large white belly, and we can just clip their fins to do genotyping. It grows right back within a week or so. And to collect eggs, what we do is we set up mating pairs in the afternoon, and then we collect the eggs in the morning. And this requires no hormones, no special treatments. They just love to produce eggs. And like here, we are seeing a mating pair. They're doing a little mating dance. You don't need to hear the sound on this, why it's coming through, but you'll see some eggs being released in a moment. There's a few. There's a few. We have to put this grid in here to let them fall through, otherwise they would eat their own eggs. So this is one egg laid from a single female. Just really amazing.
Wow.
How productive they can be. And to keep them happy and laying so many eggs, you do have to do a good job of feeding them a varied diet. They love live food. They love brine shrimp. Many of you, or some of you may know these as sea monkeys. As kids, you could do a mail order for sea monkeys and raise these in jars. This is the animal that they love to eat. We feed babies things like marine rotifers and paramecia. But you can get away with powdered or pellet food as well. As I mentioned, it is a tropical fish, so we have to keep the water pretty warm. We have to keep the rooms pretty warm. I'm scheduled to feed fish this weekend, and I will be wearing a t-shirt when I'm inside that fish room. It's pretty warm in there. Okay, so now I'd like to discuss some of the genetic tools that are available in this animal model. So people have done many forward genetic screens with this animal model, either by doing chemical mutagenesis or using retroviral insertions. People have not really been doing much of this lately, but still there are a number of labs out there doing the chemical mutagenesis. And the way this is done is that you expose a male in water with something like ethyl nitrosourea, which will cause random mutations throughout the genome. And then you allow that male to recover, and you try to mate it to as many females as possible, like 50 different females. And then you raise all those fish, and you cross them again to a wild-type fish. You raise all those fish, and now you can in-cross these to look for things like recessive mutations that would cause a phenotype in 25% of your eggs, of your progeny. Now, because, this whole scheme takes time, to do these kind of screens, you need at least a year and many people to do all the work of crossing the fish, collecting the eggs, sorting the eggs. Many labs just don't really do this unless it's a big group effort. But we are seeing fewer and fewer of these kind of screens being done simply because the NIH doesn't fund them anymore. You used to be able to propose screens among different labs and get money for that, and they stopped doing that some time ago, funding that some time ago. Okay, so for that reason, people have been turning to reverse genetic tools. And many of you may have heard of Morpholinos. They were extremely popular when they first came out. They're very easy to use. This is an antisense oligo that you inject into the one-cell stage, and then you look for a phenotype. So the way it works is the antisense oligo binds to your transcript and blocks translation, so you get no proteins. And this results in a phenotype. In this case, there are no red blood cells in this Morpholino injected fish. And like I said, they were very popular. Many people used them, but after a few years, the zebrafish community kind of realized that there were some major problems with Morpholinos. So many people saw these off-target effects of hindbrain death, small heads, small eyes, gnarly tails and stuff. And if you look at some of the publications early on, you will see groups reporting this as their phenotype because they didn't know better or they weren't so familiar with the model system. So because of these side effects, the community sort of backed away from the use of Morpholinos, and they've turned to CRISPR, which doesn't cause as many off-target effects usually. And I'll come back to CRISPRs a little bit later. Just wanted to mention one last other reverse genetic technique, which is known as mutagenesis, with the idea being that you mutagenize, like I said before, but you just sequence the DNA and you freeze the sperm away. And there's another term for this called tilling. This was done some time ago by the Sanger Institute, and they found mutations in well over 11,000 genes. And so if you don't want to do CRISPR and there's one of these mutants available, you can just go through their database and look for that and have it sent to you if you simply don't have the time to generate the knockout yourself. But sometimes it can be quicker to do the CRISPR knockout yourself. So over the years, we've collected a number of mutants just with the forward genetics approach. These are novel cadherins, unconventional myosins. Maybe some of you recognize some of these gene acronyms. They are implicated in human deafness. So, there's medical relevancy to using this animal model to study hair cell function. In some cases, we had gene duplicates where we had to use CRISPR to get knockouts of all the genes. In some cases, we also used Morpholinos to get at function of various genes. So another key feature is the ease of transgenesis in zebrafish. So we use the Toll-2 transposase system. It's fairly easy to express GFP tag or whatever kind of tag you want, tag gene. You can generate dominant negatives this way. And the way it works is that you take your plasmid of interest with a promoter and GFP, you throw in that transposase into the one cell stage, and then you raise those injected eggs, you look for a founder fish, and then you have your stable transgenic line. And here are some examples of promoters that are widely used in zebrafish. There are ubiquitous promoters that are expressed in every cell type. There are lots of tissue-specific ones. Here I'm showing you a promoter that lights up the vasculature in the developing zebrafish. Here's one that's expressed just in hair cells only. So we have hair cells in the ear, but also in the lateral line organ, which Rob brought up earlier. I'll come back to that in a moment. There are also a number of inducible promoters. There is the Crelux system in fish, but not too many labs use that. It's a little bit more work than simply trying a heat shock promoter, for example. That's probably the most popular inducible promoter used today. Okay, and then just to bring up some of the disease models that people are using zebrafish for. Well, here are some general areas of study. So most people are, trying to take advantage of that transparency during the early stages. So embryology development was, of course, one of the key areas used in zebrafish studies. Also, a lot of people then moved on to the development of various organs and tissues. They're widely used in neuroscience. I'll show you a little bit of that later. And then they're used for cancer and various disease models. And because some of you are interested in cancer, I threw in this melanoma model. So this was a study done by Len Zon's lab in which they screened 2,000 human tumors by grafting them into fish and then looking for development. And they found this one particular gene which was found to accelerate tumor growth. And then they screened all these human cell lines and found that it was indeed upregulated in 70% of those melanoma cell lines. So that's an example of the power of this model system for studying cancer. It's also used for studying toxicology and regeneration. Of particular interest, of course, is hair cell regeneration. This is a scanning electron micrograph of the face of a fish. So the throat would be down in there somewhere through the mouth. The eyes are kind of out of the picture. Here are the olfactory pits. They don't actually have a nasal passage. So fish don't inhale odors. They have this really neat trick of having motile cilia around their sensory cilia that beat in this fashion that bring in odorants and then clear them. So they sample and smell that way. Now you'll also notice these little hair, clusters of hairs here and there. These are actually kinocilia of sensory hair cells. And these little clusters are all along the surface of the fish, including going down a line of the body, and this is called the lateral line organ. And the fish uses the lateral line to sense water movements. So if a fish is near the surface of the water and a little bug lands near it, it can sense the rippling with this organ. And the sensory hair cells of this organ look very similar, if not identical, to the sensory hair cells of the inner ear. We have yet to find any major differences between these two sensory organs. And importantly, you can use the lateral line hair cells are, like our hair cells, sensitive to ototoxic drugs. Here's an example of a cluster of these hair cells treated with neomycin. All the hair cells are dead. And what's great about the fish is that, these are hair cells at the surface of the skin. All you need to do is throw the fish into the drug to get that kind of death. And you can do things like add compounds to try to block that death. And this was done by a group at the University of Washington, Ed Rubel, David Rabel. They formed this company because they had really great success with this particular compound. So the FDA has recently okayed that as something to use to protect people from hearing loss. It made it through a phase 1 clinical trial. So with oral dosing, it had a greater than 72-hour half-life. It was well tolerated, no side effects. They're on to their second phase clinical trial now. Okay, so actually once you kill all the hair cells with something like neomycin, the supporting cells will divide and give rise to new hair cells. So they just regenerate on their own. There's nothing needed and no kind of cocktail to get them to do this. So they're like chickens. And you can do other kinds of drug screens for, let's say, things that inhibit their regeneration or enhance their regeneration. So this system is a really powerful system for doing these high throughput drug screens for things that prevent autotoxicity or promote hair cell regeneration. Okay, so now I'd like to turn to using the fish as a model for research and how it might affect, something that you as ENT doctors, how it would affect your work and your research. So let's just imagine that you have this patient with hereditary hearing loss, and you do all of your usual testing and analyses, and you tell the parents that the child has sensory neural hearing loss, is actually affecting the function of the inner ear, likely like let's say the outer hair cells, and so the best treatment may be a cochlear implant for this child. But it turns out that these parents are really tech savvy, and they're reading up on things, and they're seeing stories like this. FDA approves gene therapy for inherited blindness, and they're thinking, gosh, they're already doing it for blindness. Maybe, rescuing hearing is just right around the corner. We actually want to know what the gene is that's affected in our baby. Can you please help us with this? And so you think, okay, let's do some personalized medicine. There's a couple of companies out there that are offering, sequencing of known deafness genes. So let's just try these two different panels from Otogenics or Otoscope. And you do that, but you find no mutations. And actually the parents are really disappointed. But while they're scrolling through this website on Otogenics, they come to this section on that website, and they see this option to test for novel deafness genes. And they click here, and they find this magical web page that promises them the opportunity for precision medicine, not just personalized, but precision medicine now, and which is accomplished through sequencing of the genome. So you send the DNA from the patients to Otogenics for this, and they actually find mutations. They find 2 mutations, not just 1, but 2. So you have gene X with a nonsense mutation, but there's also gene Y with a missense mutation in something that's so highly conserved in all species that it makes sense that it could be this gene as well. And just looking at these 2 candidate genes, you might think, okay, nonsense mutation. It's obvious that it should be that one. But just some words of caution about nonsense mutations, in that there's plenty of evidence that the translational machinery can sometimes read through stock codons, and there's not a problem with the expression of those genes. Or the splicing machinery can somehow recognize an exon containing a nonsense mutation and will just splice it out. This is especially prevalent in large genes with many exons, where if you just have one exon missing, it doesn't necessarily result in the loss of protein function. Then there's also the case where a nonsense mutation might lead to messenger RNA degradation. This is called nonsense-mediated decay. And in that situation, if you have a closely related gene, a parallel, it may offer genetic compensation and sort of fill in that role by that is normally provided by gene X. So, if you and I were to have our genome sequenced, we would probably find a number of nonsense mutations that would make you wonder why you're still alive. Mm-hmm. And it's probably due to, some of these reasons. So, you're stuck with this situation of two candidate genes. You go through, do some searching in the database, and it turns out that they're novel proteins. There's not much known about them. People haven't really worked on them. The expression data is sort of ambiguous. It's not clear if it's expressed in the ear or not. So you aren't sure what to say to the parents, but they're kind of relentless. And they say to you, I want you to figure this out. They actually happen to be well off. And they offer you money to do a pilot study. And, you're thinking about that money and doing that pilot study, but you're a little bit worried because these parents want an answer fairly quick, and they're not giving you a lot of money to do this. And you know that with mouse models, they're used to study human deafness, but they take time and they cost money. But you've heard about zebrafish through this lecture, and you think, okay, maybe I could try using zebrafish because they're cheaper and faster, and maybe I'll get a quicker answer to which gene is causative to the deafness in this patient. So you decide to collaborate with someone that knows what they're doing. And one of the first questions you ask is, well, which, if any of these genes are expressed in the inner ear or the auditory system? So you do in situ hybridization with RNA probes. Here's an example of a gene. It's one of the TMC genes, expressed just in sensory hair cells in the inner ear and expressed nowhere else in the fish. That's a pretty straightforward situation, but you can have expression elsewhere. So this is hair cells only. This is a gene in hair cells, but also in the brain. And then here's a gene where it's CNS only. And this might be relevant for patients where you think the defect is not in the inner ear, but it's rather a central deficit that the patient has. Okay, and so actually it turns out both are expressed in the ear, so this doesn't help you to make your decision, right? So the next thing to try is to do a CRISPR knockout. And normally people just inject one guide RNA to create a deletion in a gene. But recently it was found that if you injected multiple guides against different exons of a gene into the same egg with that enzyme that's necessary for cutting the DNA, then you can see a phenotype in that injected egg several days later. And so let's say you do your injections and then you're interested in determining whether there is any sort of deafness or behavioral deficits. Well, the easiest thing to do, oh no, why aren't you playing? It's just to tap on the dish. This is eliciting the acoustic star reflex. These are all wild types. So they're just darting around, trying to avoid that sound. Here, this is a close-up of mutants only, And you can see they have balance defects. They're popping up and landing on their heads. They're swimming in circles. So if you see this kind of defect combined with a lack of response to this, that's a pretty good sign that your gene is involved in inner ear function. Now, it might not be a black or white situation. It could be that your patient has some hearing left, it's not completely absent, so you may have to quantify the hearing loss or the vestibular dysfunction in your animal model. And we can do that by quantifying the response to acoustic stimuli using the acoustic startle reflex. We can look at vestibulo-ocular or vestibular-spinal reflexes. So for acoustic function, what we do is we put the larva into these little wells filled with water, and this plate is connected to a speaker that plays tones, in this case at kilohertz, at 156 dB, and we look for this startle reflex. And that involves, hair cell, auditory nerve, interacting with a giant escape fiber, which can then activate motor neurons. And you can quantify that response. Here is the vestibular ocular reflex. So we rotate the fish through space, and the eyes move in a reflexive pattern that corresponds to the movement of the rotating platform. And here's a video of the same exact fish. I don't know, my movies are not playing. Filmed in either dark or bright conditions. So in the dark, the eyes aren't moving in response to the platform moving back and forth. That black frame is just telling you about the position of the platform. But in the bright, those eyes are moving, bright conditions. And that's because this fish can still see, and it's seeing the movement of its environment. And this tells us that the function of the eyes is okay. But in the dark, no movement of those eyes tells us that the function of the ear is completely absent in this.
What age are these fish?
Day 5, so 5 days post-fertilization.
So these systems are mature enough that all the tests that you're doing and things are relevant and in an adult?
Yes, I mean, we haven't done these same tests in adults. Some people have done similar experiments, but it is the same circuit and reflex. That's the beauty of the fast development of the fish. All these things are in place pretty early on. Okay, so with the vestibulospinal reflex assay, we use that same moving platform, but in this case, we secure the head and we leave the tail free to move. And here's a video of that kind of response. So this fish, the platform will start moving. You'll see the water swaying a little bit, and it'll start moving its tail as that platform moves back and forth. It'll rest in between, and it'll give us a little bit of a reflexive tail movement, and so on. So this is actually using a tracking software, and with this software we can generate something like this, showing us the differences in the tail angles moving from left to right. So back and forth, back and forth. This is actually a wild-type fish. Here's an example of a mutant that responds at first, but then just stops. This back and forth is just the tail swaying back and forth because the platform is moving. So you can do that kind of test with your CRISPR mutants. Well, let's say that you have obvious behavioral deficits. Are there more? You could stop there and just say, okay, I think it's this gene based on the fact that we see a deafness phenotype. But if you're interested in exploring, why the fish are deaf and trying to explain to the parents why the patient is deaf, then you could take it a bit further and delve more into the phenotype by looking for morphological defects. For example, you can easily visualize the sensory hair cells inside the ears using transgenic lines. This is actually a line that is showing you the red the stereocilia, the actin-filled stereocilia in red, and the kinocilia in yellow. That's tubulin versus tagged actin. And there's also tubulin in the cell bodies. You can also look at like a component of the transduction apparatus in a live undissected fish. Here's the tip-link protein, proteogram 15, tagged with GFP. It's shown in the red channel. You can see whether that's properly localized in your per-sperm mutant. You can look at the synapses at the base of the cells. This is the ribeye protein tagged with GFP, so you can visualize the synaptic ribbons in live intact fish. If you thought it was a problem with synaptogenesis with the hair cells, you could look at the afferent fibers, which are known to innervate the hair cells. You can even look at efferent fibers, which modulate the activity of hair cells. So we have all these different transgenic lines to visualize the different components of the auditory system. You can also just do sort of conventional microscopy, scanning electron microscopy or transmission electron microscopy or labeling with antibodies if you want to. For those of you who are curious about, what these hair cells look like in the fish, this is a hair cell in the inner ear. It's from a macular organ. They look very much like vestibular type 2 hair cells. They don't have the calyx of a type 1, but they have sort of the basic features of vestibular hair cells that you see in other species. Here's a hair bundle, the ribbon synapses, an efferent bouton, the tip length, and then a cross-section through hair bundle, showing you the kinocilium versus the actin-filled stereocilium. Okay, well, it could be that you don't see any morphological defects because this mutation causes a functional defect. So how can you get at functional defects? Well, you can look at physiology. Some basic things to try would be to insert a recording pipette into the ear and then provide a mechanical stimulus with a glass probe and just record microphonic responses. And that's shown here in response to the step stimuli of moving that probe back and forth on the head. You can also do something like non-invasive physiological experiments. You may recognize this image from earlier. This is actually a calcium sensor that's being expressed in the sensory hair cells. So you can just, again, use a glass probe to mechanically stimulate the ear and look at activity in these without having to insert a recording pipette. What I think is really cool, let's say, your patient doesn't have that inner ear defect, it's rather a central defect. One of the huge advantages to zebrafish is that transparency and the ability to do something like brain-wide imaging. So in these experiments, this was done by Volker Bormuth's lab in France. What they've done is they've created this platform that rocks back and forth and gives a vestibular stimulus shown here, and you see the eyes moving in response to that rocking back and forth. And here, these are different planes through the brain. So here's the cerebellum, the two lobes of the cerebellum lighting up. There's a lot of activity in the optic tectum, so the vestibular system is feeding information to the visual system as well. If you go a bit deeper, this is the vestibular nucleus in the hindbrain, and these are the ocular motor nuclei lighting up in response to that rocking.
How do you visualize at different levels in an intact
So
organism?
this is actually a light sheet microscope, scanning microscope. And we actually are collaborating with Volker's lab, and they looked at one of our mutants with a central deficit, and what we're seeing in our mutant is a lack of a response in the hindbrain. So in this mutant, we know that the VOR is intact, and we actually see that the ocular motor light up, and we see activity in the vestibular nuclei near the brain. But we know that the vestibular spinal reflex is absent, and so it makes sense to us that the hindbrain, which is responsible for that motor output, is not active in this assay. So let's just sum this up by saying you actually figure out that it is gene X, it's not gene Y for various reasons. And so you can tell the parents that it's actually gene X that's causing the deafness. And you can write up the results, especially if you do some of those other experiments that I outlined that went beyond the behavioral analysis. And then you can do it all over again with the next patient. Okay, so with that, I'll end my seminar. Thanks for your attention.
It's very exciting work and very nicely calibrated for an audience of clinicians. I think that was terrific. You know, if we see human genetic patterns that aren't yet identified, but are clearly appear to be genetic or heterofamilial, is there any way we can connect these families with you to do screens in this model, or is that a bit unrealistic?
Well, I would need the clinician to do the genetic testing or go through AutoGene to figure out potential candidate genes.
Yeah.
And then I guess I would prefer that the clinician rather than the family collaborates with me.
Yeah.
Of course, you know, I'll talk to the family,
Yeah.
but I don't really want to be receiving directives from the family because, you know, anyway.
We won't talk to you, zebrafish.
I think it's totally possible to have sort of this kind of setup, and I would look forward to it because I'm kind of surprised that people haven't approached me to do this kind of work.
Take that as an invitation.
Yes.
I just saw a patient last week who has superior canal dehiscence, and her brother does as well. And it turns out there are small series of people recognizing that superior semicircular canal dehiscence has familial clusters.
Mm-hmm.
Nobody's yet done much with that. So.
But something like that is the semicircular canal, the anatomy is somewhat disrupted.
It's the inotic capsule defect, yes. Yeah.
So that would require a very long-term kind of study because that's going to be an adult phenotype.
Oh, yeah.
The semicircular canals take some time, like at least 25 days before they even become functional.
Yeah.
So that would, you could still do it, but that kind of study would just take more time simply because you have to wait at least a month before you can see your phenotype.
Any other questions or comments? Thank you, Teresa.